This is part 5 of the lectures on sexual reproduction. Sorry, not on sexual reproduction, on cell division. This slide is on sexual reproduction, and I just wanted to make sure you understood. I don't know how much you heard from the last um, lecture, but I wanted to make sure you understood some things. And one is that the process of meiosis produces gametes, and gametes, an egg is a gamete and a sperm is a gamete. So gametes are egg and sperm, and they're always haploid. Gametes are always haploid. They always have half the number of chromosomes of the regular body cells of that organism. They're always haploid. It doesn't matter which type of organism we're looking at. Always the gametes are haploid. So life cycles describe how organisms basically alternate between stages of life. So there's three main categories of life cycles. There are diploid dominant, which is most common, demonstrated by most animals, including humans, and it's the one that we're probably the most familiar with. Then there's haploid dominant, which is demonstrated by fungi and some algae. And dominant just means that the cells that form the adult organism, the organism that you recognize as that organism, like the cells that form um, a, a, I don't, I'm trying to think of a fungus. The cells that form a mushroom would be haploid. The cells that form the body cells of a dog would be haploid. And then plants exhibit an alternation of generations. They exhibit part of their life is diploid and part of it is haploid. So humans is the easiest one. What happens is this, is that you have in the sex cells of the male in the testes, um, the sex cells in the testes and ovaries divide by meiosis and produce eggs, which have 23 chromosomes and are haploid, and sperm, which also have 23 chromosomes, so they're haploid. And when the sperm fertilizes the egg, you have a zygote, which is the first cell that's going to form a new individual. And that zygote is going to be diploid. It will have 46 chromosomes, which is the addition of 23 plus 23. Now that zygote is going to undergo mitosis, until so many times until it forms trillions of cells. There are trillions of cells in an adult male and adult female. And then the process begins over and over. So the human, from the time it is a zygote, is going to be diploid dominant because all the cells are going to be diploid except the sperm and the egg. Now, here's an example of just a typical fungus. Um, which will have, I'm trying to think how to explain it to based on what we just talked about. So, okay, so you will have a fungus that mainly these little threads, they're called hyphae. All right, they're called hyphae. And the hyphae of a fungus can be either the positive mating type or the negative mating type. And if they're the positive mating type, they'll produce a positive gamete, which is haploid. And if they're the negative mating type, they'll produce negative gametes, which are haploid. And so the fungus itself, the hyphae, all the cells that form the hyphae are haploid. Because we're in the haploid region of the diagram. So I don't know how many chromosomes are in a fungus, but I do know it's probably not 23 for the haploid number, but it's some haploid number in the mating types. And so two of their cells are going to fuse. And when they fuse, now what you have in here is a zygospore, which is diploid. It's going to undergo meiosis and it's going to produce spores, and those spores will undergo mitosis. Those spores are going to be haploid, and those spores are going to undergo mitosis and form into more hyphae. Now, those hyphae can also just reproduce asexually, just clone themselves. So they can, the life cycle is haploid dominant because the fungus that you see in nature, the cells of that fungus are haploid. In plants, you have an alternation of generations, and they have used a fern here because flowering plants and like evergreen trees or trees that have cones, gymnosperms and angiosperms. Gymnosperms are the evergreens, and angiosperms are the flowering plants. They're a little bit um, less of a they're not as good of an example of alternation of generations because they have become more and more like humans in their life cycles. But this is a fern. So what we're going to look at is in the fern, the adult fern, I'll start here since this is what I know you recognize. On the underside of the leaflets of the fern are sporangia, and each of those sporangia undergo meiosis. So that means they're diploid. They undergo meiosis and produce haploid spores. Those spores land on the ground and germinate, and they grow into a gametophyte, which is a plant all on its own. It's a little plant that has a heart-shaped leaf and some rootlets. And the leaf lies flat against the ground, and it is very hard to find a fern gametophyte in nature because the ferns grow in the forest. They're usually, those leaflets are the little gametophytes usually covered up by, you know, dead leaves and that kind of thing. So, but anyway, that's what it looks like. A fern starts out as this little heart-shaped leaf that lies flat against the ground. That's a gametophyte, and all the cells in that gametophyte are haploid. Some of the cells divide by, or some of the cells produce sperm and some produce eggs. And the sperm swim to the egg. And when they do, they are now have produced a zygote, and that zygote will grow into this sporophyte. This sporophyte is eventually going to grow into the sporophyte, the fern itself. So the generation that is dominant is the sporophyte generation. The generation, but the other generation is the gametophyte generation. It alternates its life between the sporophyte and the gametophyte generation, even though the dominant generation is the sporophyte, and it is diploid. Okay, so genetic variation, crossing over, we already discussed, leads to genetic variation. Random orientation of homologous pairs, we call that independent assortment in genetics. So these are two ways that genetic variation occurs. We've discussed those already. This is just a picture of how chromosomes can randomly orient. Um, they can orient this. Okay, so this is the parent cell, and this can be the egg or the sperm, but you can get a red and a purple chromosome, a little red and a big purple, big red, little purple. But if they orient themselves like this, both red chromosomes on the left and or on the bottom, and both purple chromosomes on the top, then all you can get are two purple chromosomes or two red chromosomes in a gamete, or in a in an egg or sperm. All right, chromosome structural changes. Cytologists, which are people who study cells, have characterized numerous structural rearrangements in chromosomes, but chromosome inversion and translocation are the most common. Both are identified during meiosis by the adaptive pairing of rearranged chromosomes with their former homologs to maintain appropriate gene alignment. So what that means, what does that mean? Okay, so if you look at a normal chromosome, and these are representing genes in the chromosome, A, B, C, this is the center of the chromosome where the kinetochore is, and then D, E, and F. These are the genes. If C and D switch places and they are right side by side each other, that is called a pericentric inversion. And they're both on either side of the centromere, the um, place where the sister chromatids would be attached. If the inversion occurs with two genes that are separated from the centromere, then that's called a paracentric inversion. And those can lead to genetic diversity. Then you can have inversion pairing. When one chromosome undergoes an inversion but the other does not, one chromosome to match the other chromosome can form an inverted loop. If this one had 5, 6, 7, the six would match up, but unless you do an inverted loop, it would be 5 matching up with 7 and 7 matching up with 5. So now you have 7, 6, 5. So inversion pairing is essential to maintaining gene alignment during meiosis and to allow for recombination. So that's something that chromosomes can do to make sure they're paired up correctly. Translocations mean that a piece of DNA from another chromosome is inserted into a non-homologous chromosome. And what you can see here is these are two separate chromosomes, and this is where the piece has been inserted here, and then this is where the piece has been inserted here. There's no gain or loss of genetic information, and it can be a bad thing or a good thing. It sometimes um, it always just depends on the situation as to whether it causes a problem in the child. The karyotype, remember, is a picture of all of the chromosome pairs. And what we can tell from this karyotype is that we have a female, a biological female. And since all of the pairs only have two chromosomes, we don't see any chromosomal abnormalities. Now on pair 21, if we had another third chromosome, then that would be trisomy 21 or Down syndrome. So that child would have some version of Down syndrome. So karyotypes can help us determine the sex and also whether or not the child has a chromosomal abnormality. Non-disjunction, when non-disjunction occurs, and that's how you get these chromosomal abnormalities. So non-disjunction is failure to separate either during meiosis 1 or meiosis 2, and it ends up giving you extra or either one extra or one less chromosome than you're supposed to have. So you're supposed to have two chromosomes in the haploid egg or sperm. Well, here, because of non-disjunction, we have only one, and here we have three. And that's when the non-disjunction occurred during meiosis 2. But when the non-disjunction occurs during meiosis 1, the possibilities are either having a cell with only one chromosome each or a cell with an extra 3 chromosomes each, an extra one in each of those cells. So it's really worse if the non-disjunction happens in meiosis 1 because it will affect all of the sex cells that are formed. Monosomy and trisomy is what we call, monosomy is what we call an individual that only has one chromosome in a pair, and trisomy is when the individual has 3 chromosomes in the pair. I already told you which one corresponds to Down syndrome. There's also okay, you can also have duplications and deletions, which can be chromosomal abnormalities as well. Cry du chat, which means cry of the cat, is associated with nervous system abnormalities, and it's a deletion of most of 5p, which is the small arm of chromosome 5.